السلام عليكم. في محاضرة الليلة إن شاء الله هنتكلم عن النيوكليك أسيد and protein extraction. آه، دي واحدة من التوبكس اللي هتشتغلوا عليها في اللاب إن شاء الله، أو يعني هشرحها لكم بتفصيل أكتر، شغل عملي في اللاب، فالناس تركز إن شاء الله. طيب الاكستراكشن، عملية الاكستراكشن للنيوكليك أسيدز الدي ان اي والار ان اي والبروتين دايما بتكون هي starting point in any molecular biology research or study. يعني أول حاجة بيعملوا شنو؟ بيعملوا extraction للبروتين أو النيوكليك أسيد اللي هم عايزين يعملوا عليه تيست. الاكستراكشن بيكون بيتم أو sources of these materials ممكن يكون من living organism, tissues, cells, viruses, particles, or even biological samples. فممكن كلها ممكن نعمل extract لبروتينز أو DNA أو RNA for molecular biology study. طيب what is the purpose of extracting؟ زي ما قلنا releasing of the nucleic acid or protein from the cell to be available as a pure compound for testing. لازم يكون free from contamination، يعني ممكن نعمل extraction لأنه السامبل ممكن يكون contaminated، فلازم نعمل له extraction or في الحالة دي يسمى purification. في الأخير إحنا عايزين نستخلص المولكيول اللي إحنا عايزين نشتغل عليه أو الكومباوند اللي إحنا عايزين نشتغل عليه لازم يكون pure علشان يكون available for testing زي ما قلنا. Quality and integrity هما the main issue that or it should be our concern while we are extracting or purifying nucleic acid or protein. Both are very sensitive like many conditions, changing conditions like pH, temperature. كل ما يعني إذا هي nucleic acid أو nucleic acids أو البروتينز لازم نكون يعني منتبهين إنه during the extraction or purification we should maintain the quality and the integrity of these compounds. طيب نبدأ بالnucleic acid extraction methods. طيب the choice of the extraction method, suitable extraction technique for nucleic acid depends on the target nucleic acid إذا إحنا عايزين نشتغل على DNA or RNA, the source organism من bacteria or fungal or human cell or starting material. إذا هنبدأ بتشو leaf ممكن يكون plant حتى. desired result that the outcome should be like a quantity. We are just looking for quantity of that nucleic acid or purifying or other reason why we are doing extraction and application. For what reason we are extracting this compound. So these are the criteria by which we choose the suitable extracting technique. For protein extraction, we need to extract the protein in order to study their unique characteristics, including size, shape, and functions. And for extracting the protein, we need to free the protein from the cell. How? By lysing the cell or by using lysing agent like detergents, chemicals that could lysis the cell. Lysis the cell means break down the cell membrane, and release its content, including proteins or nucleic acid or any other components inside the cell. So shearing force also, if we apply some kind of force to the cell could rupture and release its content. Also salting out, we can use some chemicals that uh, lead to salt the release the protein. The method called salting out, so the protein will be released from the cell. The other method is pressure, applying pressure with sudden change in the pressure. So also the cell will rupture and release its content. So the whole idea from this is to break the membrane surrounding the cell to allow protein to escape. This is the first step. Some factors we need to consider when we handling the protein during the purification or extraction. First, the whole process should be conducted at low temperature, around 4 degrees Celsius, because protein is sensitive to heat. Most of proteins they can naturate at high temperature. Also, we should maintain buffer condition appropriate to the protein that we are working on it, because protein also sensitive to change in pH. Also, the solvents we are using, for example water, if we are using some solvents or water, they should be pure, because any contamination, for example like microorganisms or enzymes, they could also degrade the protein. Also, some solvents or buffer contain protein inhibitors, so these inhibitors could also attack the protein and affect its nature. Reducing agents are required during the purification or extraction process to prevent oxidation of the proteins. Also, the storage condition for the protein after the protein being extracted or purified, we should maintain proper storage condition, especially temperature. It should be stored in fridge to increase their half-life. There's many methods used for extracting DNA, RNA, and proteins. This method can be divided into two main categories, which is solution-based or column-based protocols. So all the methods is coming under these two categories. We start with the nucleic acids extraction methods. There's three main goals from each extracting method. First, preparation of cell lysate or lysing the cell. We can use mechanical disruption, for example grinding. We can grind the cell or the source of the nucleic acid. We can grind the source, then it will release the nucleic acid. Other methods is chemical treatment. We can treat the cell, for example, with chemical reagents like detergents, then the nucleic acid could escape from the cell, so the cell will rupture and release nucleic acids. Or enzymatic digestion, also we can use some enzymes can be used to lysate the cell. The second goal is to activate the cellular nuclease, because cellular nucleases also could attack or denaturate the nucleic acids. third, separation of the desired nucleic acid from the cellular debris. So the separate the nucleic acid from the other component of the cell lysate. So the waste of the cell lysate, other than nucleic acid, is considered as a waste. So we need to separate the nucleic acid from the waste of cell lysate. So we will start with the conventional methods. Traditional methods. The first method is phenol chloroform extraction. So it's by phase emulsion or solvent system. They're immiscible solvents. So due to the difference in the solubility, the components of the cell lysate will separate. So how? In the hydrophobic layer, there will be the cell debris. As we said, as I just mentioned, that anything other than the nucleic acid is considered as cell waste. So all the cell component, after lysing the cell, all the cell component will be in the hydrophobic layer at the bottom of this system, extraction system. In the hydrophilic layer at the top, we will find our nucleic acid. Then we will remove that layer and we will add ethanol, cold ethanol. Then the DNA will precipitate, or the nucleic acid will precipitate. Then we can separate by centrifugation. So we centrifuge, then we can separate, discard the supernatant or the liquid part, then we will get the nucleic acid. So the second method from the traditional method is alkaline extraction method. So in this this method mainly used for extracting plasmid DNA, so the bacterial DNA, by the use of sodium dodecyl sulfate, or the SDS. This solvent, or this reagent will denaturate the high molecular weight DNA. So when we add or treat the cell with this reagent, it will break down all the high molecular weight DNA and just leave the circular DNA, which is the plasmid. اتكلمنا عنها كتير، قلنا البلازميد هي circular DNA. طيب بعد كده، طيب زي ما قلنا عندنا، نفترض انه عندنا السيل لايسيت، بنضيف الرياجنت اللي هو ال SDS. SDS بيعمل بريك داون لكل ال high molecular weight nucleic acids and leave the plasmid, the circular nucleic acid, intact. Then that nucleic acid, the plasmid, then can be recovered by centrifugation also. طيب دي قلنا بنسميها alkaline extraction method. The third one is CTAB extraction method. This one mainly used for extracting nucleic acids from plant sources. First, we need to free the nucleic acid from the cell. How? We freeze the plant material using liquid nitrogen first. Then we grind this material, frozen material, we grind it to break down the cell wall. Then why we freeze it first? To keep to get access to the nucleic acid, but at the same time freezing the cell will keep all the harmful materials inactive, like chemicals and enzymes inside the cell that could harm the nucleic acid. So we will freeze them then we will grind the cell or the material to break the cell wall and free the nucleic acid. So then by adding the reagent CTAB, it will allow the nucleic acid to precipitate, and other protein and natural polysaccharide will be in solution. So the reagent will only precipitate nucleic acids. Then the insoluble particles, then to collect the nucleic acid after precipitation, it can be obtained by centrifugation. And if we also want to collect the soluble proteins, then we can mix with chloroform, then it will precipitate, then we can collect it. So both can be collected, the nucleic acids and soluble proteins. The nucleic acid can be purified and stored in distilled water, purified or sterile distilled water. لو شايفين هنا انه اغلب الستبس almost same، اغلبها almost same. عندنا cell lysate، عندنا releasing the nucleic acid from the cell, then how we can separate from other components. كل واحدة ممكن يكون عندها principles معينة ومختلفة، لكن في الأخير الكونسبت واحد، وحتى الستبس كمان هي واحدة. زي ما قلنا في الأول هم inactivation cell lysis. في الأول حاجة free the component or the nucleic acid. The second one is deactivation, and then purifying the nucleic acid. طيب بالنسبة لل how the CTAB reagent do its work. So it lyses the cellular membrane first, okay? Then second, it will bind to the membrane components such as phospholipids, lipoproteins, polysaccharides. By binding to them, it will inhibit them from precipitate. So that's why the addition of this reagent will only allow DNA to precipitate, only nucleic acids to precipitate. Other than that, the other components, cell components, will be in solution, soluble in solution. Then we can separate them easily. We can centrifuge, separate the DNA, and from the liquid also we can separate the soluble materials by adding chloroform. Then they will precipitate, then we can centrifuge and separate them. Type the fourth method from the traditional methods is gradient centrifugation. So we create two different gradients within the system, then similar to the first method. So then we mixed our cell lysate in the system, distribute all cell components in the system, then the separation will be occur due to the different between the two gradients of the system. How this will happen? Okay, first, this method is very complicated, expensive and time consuming. So and also it require large amount of bacterial culture. So it's not suitable for mini preparation of DNA. It's only suitable for large scale preparation of DNA because it's very expensive, complicated, and require large amount of bacterial culture. So first زي ما قلنا بيكون في two gradients. The first one is due to ethidium bromide, and the second one is due to cesium chloride. These two chemicals will create two different gradients within the system. So when we mix the cell lysate, the closed circular molecules, which is it refers to the plasmid, DNA plasmid, will be accumulated at the lower density region. In this case will be the CSCL region. Then other components of the cell lysate will be in the ETBR region. Then this region will remove by using appropriate hydrophobic solvent. So we can add hydrophobic solvent, then the ETBR region will dissolve in the hydrophobic solvent, then we can separate them. Then we will then there will be only the hydrophilic region containing our plasmid DNA. We can precipitate it with alcohol. Again, precipitate with alcohol and separate it. طيب نجي بعد كده more advanced method than the traditional method. The first class is, it's a big class coming. They will be under it different methods, sub methods. The first class is solid phase nucleic acid extraction. So this system consists of solid phase that is responsible for the separation of the nucleic acid. It's a quick and efficient compared to the traditional methods. The nucleic acid will be absorbed during the extraction method. How it can be absorbed? It could be due to hydrogen binding interaction. It could happen. Nucleic acid form hydrogen bonding with the solid phase, or due to ionic exchange, or due to affinity and size exclusion. هنشوف كل واحدة مثال لها. طيب mainly this kind of extraction performed by using spin column and operating under centrifugal force. All of them perform under these conditions. The solid phase mainly or commonly used the silica phase and the anion exchange carriers. This mainly solid phase are used for this kind of extraction. So the steps is, first we need the cell lysis, nucleic acid adsorption will happen, then we will wash, and then elute. Elute means if our target nucleic acid is attached to something, maybe ion or anion, maybe the solid phase, maybe any other thing it's attached to, so we need to separate from that other material. هنشوف كمان في examples. طيب إذا تكلمنا overall in general, the concept behind the solid phase extraction method is how the separation will happen. So لو قلنا بيكونوا بالبلو ديل هما السوليد فيس من ناحيتين، من هنا ومن هنا. معلش طيب من هنا ومن هنا. طيب هما hydrophilic surfaces, so it will only attract the nucleic acids. So nucleic acid will be absorbed to the nucleic acid will be adsorbed to the solid phase surface. Other materials will be seep out of the membrane. يعني لما نضيف السيل لايسات to the system will catch the DNA or RNA and other materials will be released out from the system. That's it. Then we need to wash to make sure everything is other than the DNA and RNA is went out from the system. Then elution, we need to separate our DNA and RNA from the solid phase. ده شكل السيليكا purification matrix بتكون شكلها كده. بنضيف كل المواد هنا وبعد كده بيحصل ال DNA وال RNA بيكونوا attached to this part and other materials will be coming out from this hole. بعد كده بنعمل washing several times. بعد كده محتاجين نفصل our DNA and RNA from this part silica part. طيب one of the subclass for the solid case solid phase extraction method is magnetic bed based nucleic acid purification, in which the separation is based on affinity binding with magnetic separation. So it's affinity binding, magnetic separation. The nucleic acid will be linked or tagged with magnetic molecule, then magnetic field will be applied to the system. Then any material other than the DNA and RNA or other than nucleic acids, it will be washed out because it is not bind to magnetic molecule, so it will be washed out. Then we need to separate our targeted nucleic acid from the magnet molecule during the elution step. يعني الفرق انه هنا بنعمل tagging للtargeted nucleic acid بمagnet molecule. بعد كده we apply magnetic field. بعد كده بيحصل attraction for this magnetic molecule to the magnet. بنعمل washing. كل حاجة ما attached to the magnet it will be washed out. بعد كده في مرحلة elution بنفصل nucleic acid or from the magnetic material. Anion exchange also utilize silica beds, which consist or contain large bore size and high charge density. So it's based on interaction between positive charge diethyl amino ethyl cellulose DEAE group on the surface of the beds with the negative charge phosphate of the DNA backbone. يعني السيستم positively charged, it will make interaction force between the system and the DNA. How? نفترض زي ما شايفين هنا البيد silica bed is positively charged. We will add our sample contain negatively charged molecules is the nucleic acid and positively charged molecules are other cell components or protein. So then we add salt buffer. When we add medium salt buffer, it will only release the positive charge molecules, which is anything other than nucleic acids. Nucleic acid will be attached with the silica bed. Then when we add high salt solution, then we can separate our nucleic acids. So impurities such as protein and RNA are washed from the resin by using medium salt buffer. Then DNA remains bound until eluted with high salt buffer. طيب دي اللي هي anion exchange material or method. طيب نجي بعد كده للبروتين extraction methods. خلصنا من النيوكليك acid extraction methods, نجي للبروتين extraction method. First step is again cell lysis to release the protein from the host cell in a soluble form. اللي هي soluble form عشان كده بعدين ممكن أغلب الميثودز هتكون chromatography عشان نعمل له separation. نبدأ بالion exchange chromatography based on surface ion charge. So ion exchange based on the surface charge. في الحالة دي بيكون كمان عندنا bed or surface, in this case is cellulose bed اللي هو لونه بالرمادي ده بيكون negatively charged. بعد كده عندنا sample cell lysate بيكون different molecules with different charge عشان كده الألوان مختلفة. هنا بيوضح لنا إنه positively charged ions of increasing strength، يعني الأخضر الغامق هي less positive, medium positive and highly positive. أوكي، فعندنا هنا السيستم negative معناتها the less positive will be attached. Less positive will attach to the negatively charged bed. sorry العكس، أنا آسف. دي هي more positive, medium and less positive. فالmore positive will be attached or attracted to the negatively charged bed, then the medium, then the negatively or less positive it will be washed out لأنه ما في attraction. لأنه opposite charge there will be no attraction, only negative and positive attract. And so if it's more positive will be attract strongly, then medium positive less attraction, then very less positive almost no attraction. That's why it will separate first. إن شاء الله يكون واضح وما أكون لخبطتكم. طيب gel filtration, second method in protein extraction. Gel filtration chromatography, it depends on the size, so it separates protein according to the molecular size and shape. So proteins will not bind to any surface. The separation only depends on size and shape. So large molecules will be pass faster than small molecules. كيف هنشوف بعد شوية. Small molecules will enter tiny holes in the matrix and access more of the column, so take more time to run out of the. الكلام ده معناه شوية. إذا افترضنا إن إحنا عندنا السيستم من فوق، افترض لما نضيف السيل لايسات اللارج molecules ما بتدخل في الفتحات دي، ما بتدخل هنا، فمباشرة بتمر بينهم وبتطلع out of the system. أما السمول molecules بتدخل هنا فبتاخد وقت أكتر. الهولز دي ما هي straight، فالحاجة اللي بتدخل فيها بتاخد وقت أطول عشان تطلع. عشان كده large molecule will be released or separated first or faster than the small molecules. فهي كل عملية السيبريشن هي is depend on the size and shape. Advantage of this method that the molecule that's sensitive to pH change, to concentration of metal ions, to harsh environmental condition, this method is suitable for this kind of molecules because there will be no use of chemicals, no use of any kind of change in pH, and also no harmful environmental condition. Only separation is dependent on the size, different in size. Affinity chromatography, there will be interaction in this method, will be interaction between protein and solid phase. The protein will have high affinity to certain functional group on the solid phase. Mainly these functional groups is related to some molecules called ligands. So these ligands will attach to the protein by certain functional group. So then the protein will be attached to the solid phase. Other not attached proteins or not attached molecules will be washed out from the system. So the bound proteins will be eluted after the separation. The protein will be eluted from the surface. That means separated from the solid surface by solution containing high concentration of soluble form of the ligand. So بنفصل البروتين من السيرفيس ب solution containing high concentration of soluble form of the ligand. هنا عندنا اللايجنز اللي هي ممكن تستخدم في separation of the proteins، وهنا البروتينز اللي هي ممكن تفصل عن طريق اللايجنز. طيب زي ما قلنا دي اسمها الافينيتي كروماتوجرافي. طيب بعد كده نجي للجل الكتروفوريسيس، ده تكلمنا عنها تقريبا المحاضرة اللي فاتت. فللدايرين معلومات او تفاصيل اكتر يرجعوا المحاضرة اللي فاتت، لكن هنا او اللي قبل اللي فاتت هنا هنمر سريع عليها. هي depends on size and charge. So there will be a gel contain pores, a definite size of pores. So then the protein will be separated according to their size. So that means large proteins will be migrated slower than the small proteins, because the size of these pores is fixed. So small proteins will go faster, but large molecules need time to pass through these pores. How they pass? These molecules should be charged molecules. The first place should be charged molecules. Then when we apply electric charge to the gel, to the porous gel, these molecules will be migrated towards opposite charge electrodes. So there will be two electrodes in the system. One is negative, one is positive. So if the protein is negatively charged protein, so it will migrate towards the positive charge electrode, and vice versa. So if the protein is positive, it will migrate towards negative charge electrode. So it's based on the size and the charge, both. So the separation here based on the size and charge. زي ما قلنا اللي عايزين تفصيل أكتر ارجعوا لمحاضرة الجل electrophoresis. طيب احنا هنا بنتكلم عن purification or separation. So if we separate these molecules by the gel, so these molecules will be stuck in the gel. So how we can separate them or collect them from the gel? So we start with the sample from the source, then gel electrophoresis for separation. Then we will take the band. Which band is contain our protein? We should know which band is contain our protein, so that we will scratch this band from the gel, or cut it from the gel, and then dissolve it in suitable solvent. Then we will obtain our protein solution. Then we can go for analysis. So this is how we can separate our protein from other proteins or other cell content or lysate content. So this method we separate the content or constituent, or we will separate all the components of the cell lysate according to the size and charge. So each band will represent one compound. So if for example this band is for our targeted compound or protein, then we have to cut it out, dissolve in suitable solvent, and go for analysis. طيب بعد كده نجي للimmunoblotting technique of extraction. It's also used for identification and characterization. This method involves first extracting the cell lysate by SDS page. It's electrophoresis also. Electrophoresis, one kind of electrophoresis, gel electrophoresis. Then transfer to nitrocellulose filter. هنشوف بعد شوي كيف. Incubate with oligonucleotide probes. Then go for analysis. Sometimes we use antibodies instead of probes. Problem of this method is طيب الفكرة هنا قريبة جدا للافينيتي chromatography، انه بيكون البروتين عنده affinity to bind with 13 probe. احنا بنجيب البروب ده وبنخليه في النيترو سيلولوز فلتر بعد ما نعمل ترانسفير لل افضل ما اشوف الصورة. طيب هنا بيكون عندنا الكتروفوريسيس جل بعد ما عمل لنا separation لكل الكومبوننتس within the cell lysate. بنجيبه هنا and we install it in the system. This system immunoblotting system. Then the system contain nitrocellulose, okay, nitrocellulose gel. This is we need to transfer the all the proteins from the gel to the nitrocellulose sheet. So here in this step we will transfer after we incubate them together. Here هنا نعمل لهم incubation together, the gel coming from the electrophoresis with the nitrocellulose sheet. Then all the materials from the gel will be transferred to the sheet. Then we will add our antibodies or probes to the sheet. These probes or these antibodies have the ability to selectively bind to our targeted protein that we are extracting or identifying or characterizing. So then we add another set of antibodies. Then we can, after we wash or runs our sheet through certain buffers, then we can get only the band for our targeted protein, because it's the only compound that bind to our probes or antibodies. That's why it's called immunoblotting, because they will be binding between antibodies and our compound. It will bind to its complementary antibodies. Okay, there will be assignment in this course. You are required to submit an assignment from 3 to 4 pages about automated extraction or purification methods or system for DNA, RNA and proteins. And submission should be soft copy PDF file to my email, and the deadline will be 20th of January. That's mean after the final exams، يعني تقريبا 4 أيام بعد الفاينل exams. تمام، وكده بكون المحاضرة خلصت وباقي لنا محاضرة. أنا بالنسبة للجزء تبعي باقي لنا محاضرة واحدة، أما الدكتور علي عنده محاضرتين إن شاء الله غالبا بكرة نعمل لهم أبلود، وكده بنكون خلصنا. بعد كده بنكون باقي لنا بس البراكتيكال. طيب السلام عليكم.